Good day students. I will be talking about the proteins. By now you are familiar with the proteins and how they are made up of. I will be talking about the various conformations of the proteins or hierarchy in the protein structures. There are 4 levels of protein structures: primary, secondary, tertiary and quaternary. Primary structure is the one which tells you about the sequence of the amino acids, and sequence is essential for the proper physiological function. Another importance of the primary structure is that it tells you the evolutionary history of the proteins also. The two proteins having the similar sequence suggests that they have originated from the common ancestors. Now, the sequence also plays an important role in physiological or biological activity. For example, there are two hormones, one being the oxytocin and another being the ADH. These two differs only in 2 amino acids, but this difference is responsible for the diverse function of these two hormones. Oxytocin is responsible for uterus contraction and the milk production, whereas ADH is responsible for the retention of water in kidney. Now, another important example which tells the importance of sequence is the disease called sickle cell anemia. In sickle cell anemia, what happens? There is a replacement of one amino acid, that is glucine, at the position 6th to valine, and this single change in the amino acid can lead to the sickle cell anemia. So, this strongly suggests the importance of primary structure or sequence of amino acids in the proteins. Now the second level of conformation is the secondary structure. Secondary structures refers to the local conformation of some part of the polypeptide, and the two main types of secondary conformations are alpha and beta helix, and the third one is the random coils. These structures are predicted by Paul and Corres in 1951 using X-ray crystallography. Now coming to the alpha helix. Alpha helix is a right-handed or a clockwise coiled structure. It is a rod-like structure. Tightly coiled polypeptide main chain forms the inner part of the rod and side chains extends outwards in the helical array. The helix is stabilized by hydrogen bonds between the amino and the carbonyl group of the main chain. The carbonyl group of each amino acid is hydrogen bonded to the amino group of the amino acid that is situated 4 residues ahead in a linear sequence. All the main chain CO and NH groups are hydrogen bonded. There are 3.6 amino acids per turns. Amino acids spaced 3 and 4 apart in the linear sequence are spatially quite close to one another in a helix, whereas amino acids 2 apart in a linear sequence are situated on the opposite side of the helix and so are unlikely to make contact. In alpha helix, the helix has a pitch of 5.4 angstrom and the product of translation is 1.5 angstrom apart, and the number of residues per turn is 3.6 amino acids. Now there are the 5 different types of constraints that affects the stability of an alpha helix. These are electrostatic repulsions, the bulkiness of adjacent R groups, the interactions between the amino acid side chains spaced 3 and 4 residues apart, the occurrence of proline and glycine residues. As we all know that proline is not the amino acid but amino acid. Wherever the proline is present, we can see a nick or a folding in a protein chain. And the 5th one is the interaction between the amino acid residues at the ends of the helical segment and the electric dipole inherent to the alpha helix. Now the second one is beta plated sheets. These are the extended stretch of amino acids and these are called as beta strands. There are 2 types of beta parallel sheets, beta sheets, first being the parallel and the second is the anti-parallel. If the end terminals of the adjacent strands are on the same side, it is called as parallel beta sheet, and if they are on the opposite sides, they are called as anti-parallel sheets. And there are various forces which are responsible for holding the beta strand structures, and mainly being the hydrogen bonds, and it is formed between the adjacent strands. The third structure being the random coil. Random coil has a fixed structure within a given protein and is commonly called as connecting loop region, and its structure is determined by the bonding of side chains. Now the third level of conformation is the tertiary structure. Tertiary structure is formed when the secondary structures fold and pack together, and it gives the globular shape mainly. Tertiary structures are stabilized by bonds between the R groups, that is the side chains. Many proteins are organized into multiple domains which are compact globular units and are connected by a flexible segment of the polypeptide. Each domain contributes a specific function to the protein. Different proteins may share similar domain structures, that is kinase, cysteine rich or globin domains. There are 5 different types of bonds which stabilizes the tertiary structures: hydrogen bonds, van der Waals interactions, hydrophobic interaction, ionic interactions, and disulfide linkages. And the fourth and the last conformation in the protein structure is the quaternary structure. Quaternary structure dictates the association of more than one polypeptide. Each unit of this protein is called a subunit, and the protein is an oligomeric protein. Subunits can be identical or different. The protein is homopolymeric or heteropolymeric. Disulfide bonds usually stabilizes the oligomer. Now the protein undergoes the assisted folding. It requires certain proteins for its folding, and mainly chaperones are responsible for the correct folding of the proteins. In addition to this, there are certain other proteins responsible for the folding of the proteins, and these are protein disulfide isomerase and peptide prolyl cis trans isomerase. Now protein can be denatured also, and denaturation is the property when it loses its three-dimensional structure, and it is responsible, and this causes the loss of function, and this is the denaturation. Protein can be denatured by heat, by extremes of pH, by certain miscible organic solvents such as alcohol or acetones, by certain solutes such as urea or guanidine, hydrochloride, or by detergents. Each of these denaturing agents represent a relatively mild treatment in the sense that no covalent bonds in the polypeptide chains are broken. Now the last topic in this protein is the isolation of proteins. Proteins can be isolated from a given mixture or from the cells, and two main techniques used for separation of proteins are electrophoresis and mass spectroscopy, and I will be talking mainly about the electrophoresis. Electrophoresis is the movement of any molecule under the influence of current. Now the protein can be separated on the basis of their charges as well as on the basis of their molecular weight. When they are separated according to their charges, the technique is called as isoelectric focusing, and when they are separated according to their molecular weights, it is called as SDS, polyacrylamide gel electrophoresis. And when these two techniques are combined together, it is called as two-dimensional electrophoresis, and usually 2D electrophoresis is employed for the separation of the proteins. Once the proteins are separated by the electrophoresis, they are characterized by the mass spectroscopy. And I think with this we have covered the proteins. Thank you.